Hello everyone, myself Kirti. I am a student of BSc Chemistry Honors final year. This is an audio recorded lecture on the topic drugs and their classification. So the first topic is drugs and their classification. Drugs are chemicals of low molecular masses, that is 100 to 500 units. These interact with macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis, prevention, and treatment of disease. Most of the drugs used as medicines are potential poisons if taken in doses higher than those recommended. Use of chemical for therapeutic effect is called chemotherapy. Next is classification of drugs. Drugs can be classified mainly on criteria outlined as follow. First is on the basis of pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, analgesics have pain killing effect, antiseptics kill or arrest the growth of microorganisms. Second type of classification is on the basis of drug action. It is based on the action of a drug on a particular biochemical process. For example, all antihistamines inhibit the action of the compound histamine, which causes inflammation in the body. There are various ways in which action of histamines can be blocked. Next type of classification is on the basis of chemical structure. It is based on the chemical structure of the drugs. Drugs classified in this way share common structural features and often have similar pharmacological activity. For example, sulfonamides have common structural features. Next classification is on the basis of molecular targets. Drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action on targets. The classification based on molecular targets is the most useful classification for medicinal chemists. Next topic is drug target interaction. Macromolecules of biological origin perform various functions in the body. For example, proteins which perform the role of biological catalyst in the body are called enzymes. Those which are crucial to communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acid have coded genetic information for the cell. Lipids and carbohydrates are structural part of the cell membrane. We shall explain the drug target interaction with the help of enzymes and receptors. Next is enzyme as drug targets. First is catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how do enzyme catalyze the reaction. In their catalytic activity, enzyme perform two major functions. The first function of an enzyme is to hold the substrate for a chemical reaction. Active sites of enzyme hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates bind to the active site of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waals interaction, or dipole-dipole interaction. The second function of an enzyme is to provide functional groups that will attack the substrate and carry out chemical reaction. Next is drug enzyme interaction. Drug inhibit any of the above mentioned activities of enzymes. These can block the binding site of the enzyme and prevent the binding of substrate or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. Drugs inhibit the attachment of substrate on active site of enzymes in two different ways. First way is that drugs compete with the natural substrate for their attachment on the active sites of enzymes. Such drugs are called competitive inhibitors. Second is that some drugs do not bind to the enzyme's active site. These bind to a different site of enzyme which is called allosteric site. This binding of inhibitor or allosteric site changes the shape of the active site in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is a strong covalent bond and cannot be broken easily, then the enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. Next is receptors as drug targets. Receptors are proteins that are crucial to body's communication process. Majority of these are embedded in cell membranes. Receptors protein are embedded in the cell membrane in such a way that their small part possessing active site projects out of the surface of the membrane and opens on the outside region of the cell membrane. This is the figure which show receptor protein embedded in the cell membrane. The active site of the receptors open on the outside region of the cell. In the body, message between two neurons and that between neurons to muscles is communicated through certain chemicals. These chemicals, known as chemical messengers, and received at the binding site of receptor proteins. To accommodate a messenger, shape of the receptor site changes. This brings about the transfer of message into cell. Thus, chemical messengers give message to the cell without entering the cell. This is the figure. Figure A shows receptor receiving chemical messenger. Figure B shows shape of the receptor changed after attachment of messenger. And figure C shows receptor regains structure after removal of chemical messenger. There are a large number of different receptors in the body that interact with the different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor site and inhibit its natural function are called antagonists. These are useful when blocking of message is required. There are other type of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there is a lack of natural chemical messenger. Next is therapeutic action of different classes of drugs. In this section, we shall discuss the therapeutic action of a few important classes of drugs. First is antacids. Overproduction of acid in the stomach cause irritation and pain. In severe cases, ulcers are developed in the stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminium and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of even more acid. Metal hydroxide are better alternative because of being insoluble. These do not increase the pH above neutrality. These treatment control only symptoms and not the cause. Therefore, with these metal salts, the patients cannot be treated easily. In advanced stages, ulcer become life-threatening and its only treatment is removal of the affected part of the stomach. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical histamine stimulates the secretion of pepsin and hydrochloric acid in the stomach. The drug cimetidine was designed to prevent the interaction of histamine with the receptor present in the stomach wall. This resulted in release of lesser amount of acid. The importance of the drug was so much that it remained the largest selling drug in the world until another drug, ranitidine, was discovered. These are the structures of histamine, cimetidine, and ranitidine. Next classification of drugs is antihistamines. Histamine is a potent vasodilator. It has various functions. It contracts the smooth muscles in the bronchi and gut and relaxes other muscles, such as those in the wall of fine blood vessels. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Synthetic drug bromophenramine and terifenadine act as antihistamines. They interfere with the natural action of histamine by competing with histamine for binding sites of receptor where histamine exerts its effect. Now, the question that arises is, why do above mentioned antihistamines not affect the secretion of acid in stomach? The reason is that anti-allergic and antacid drugs work on different receptors. Next is neurological active drugs. First, neurological active drugs are tranquilizer. Tranquilizer and analgesics are neurological active drugs. These affect the message transfer mechanism from nerve to receptor. Tranquilizer are a class of chemical compound used for the treatment of stress and mild or even severe mental disease. These relieve anxiety, stress, irritability, or excitement by including a sense of well being. They form an essential component of sleeping pills. There are various types of tranquilizer. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that plays a role in mood changes. If the level of the noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situation, antidepressant drugs are required. These drugs inhibit the enzyme which catalyzes the degradation of noradrenaline. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for long period of time, thus concentrating the effect of depression. Iprazolam and phenazine are two such drugs. These are their structures. Some tranquilizer, namely chlorodiazepoxide and meprobamate, are relatively mild tranquilizer suitable for relieving tension. Equanil is used in controlling depression and hypertension. These are the structures given of the above drugs. Derivatives of barbiturate acid, veranol, amytal, nambutal, luminal, and second constituent an important class of tranquilizer. These derivatives are called barbiturates. Barbiturates are hypnotic sleep producing agents. Some other substances used as tranquilizer are Valium and serotonin. These are the structures of veronal, Valium, and serotonin. Analgeses. Analgeses reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination or paralysis or some other disturbances of nervous system. These are classified as follow: narcotic drugs and non-narcotic drugs. Non-narcotic or non-addictive analgesics. Aspirin and paracetamol belong to the class of non-narcotic analgesics. Aspirin is the most familiar example. Aspirin inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain, such as that due to arthritis. These drugs have many other effects, such as reducing fever, antipyretic, and preventing platelet coagulation. Because of its anti blood clotting action, aspirin finds use in prevention of heart attacks. Narcotic analgesics, morphine, and many of its homologs, when administered in medical doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions, and ultimately death. Morphine narcotics are sometimes referred as opiates since they are obtained from opium poppy. These analgesics are chiefly used for the relief of postoperative pain, cardiac pain, and pain of terminal cancer and in childbirth. These are the structures of morphine, heroin, and codeine. Next is antimicrobials. Disease in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. An antimicrobial tends to destroy or prevent development or inhibit the pathogenic action of microbes such as bacteria, antibacterial drugs, fungi, antifungal agents, virus antiviral agents, or other parasites, antiparasitic drugs selectively. Antibiotics, antiseptics, and disinfectant are antimicrobial drugs. Now we discuss about antibiotics. Antibiotics are used as drugs to treat infection because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms such as bacteria, fungi, and molds that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in synthesizing some of the compounds that were originally discovered as product of microorganisms. Also, some purely synthetic compounds have antibacterial activity and therefore definition of antibiotic has been modified. An antibiotic now refers to a substance produced wholly or partially by chemical synthesis, which is which in low concentration inhibits the growth or destroy microorganism by intervening in their metabolic processes. The search for chemicals that would adversely affect invading bacteria but not the host began in the 19th century. Paul Herrlich, a German bacteriologist, conceived this idea. He investigated arsenic based structure in order to produce less toxic substances for the treatment of syphilis. He developed the medicine arcefamine known as salvarsan. Paul got Nobel Prize for Medicine in 1908 for this discovery. It was the first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on the bacteria which cause syphilis is much greater than on human beings. At the same time, Paul was working on azodie also. He noted that there is similarity in structure of salvarsan and azodie. The arsenic linkage prevent in arcefamine resemble to the nitrogen linkage present in azodie in the sense that arsenic atom is present in place of nitrogen. He also noted tissue getting colored by dye selectively. Therefore, Paul began to search for the compounds which resemble in structure to azodie and selectivity bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial agent, Prontosil, which resemble in structure to the compound salvarsan. Soon it was discovered that in the body, prontosol is converted to a compound called sulfanilamide, which is the real active compound. Thus, the sulfa drugs were discovered. A large range of sulfonamide analogous was synthesized. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the antibacterial property of a penicillium fungus. Isolation and purification of active compounds to accumulate sufficient material for clinical trials took 13 years. Antibiotics have either sedal, that is killing effect, or a static effect on microbes. A few examples of two type of antibiotics as follow. Bacterial sedal are penicillin, ofloxacin, etc. And bacteriostatic are chloramphenicol, tetracycline, etc. The range of bacteria or other microorganisms that are affected by a certain antibiotic is expressed at its spectrum of action. Antibiotics, which kill or inhibit a wide range of gram positive and gram negative bacteria, are said to be broad spectrum antibiotics. Those effective mainly against gram positive or gram negative bacteria are narrow spectrum antibiotics. If effective against a single organism or disease, they are referred as limited spectrum antibiotics. Penicillin G has a narrow spectrum. Ampicillin, amoxicillin are synthetic modifications of penicillins. These have broad spectrum. It is absolutely essential to test the patients for sensitivity, that is allergy, to penicillin before it is administered. In India, penicillin is manufactured at the Hindustan Antibiotics in Pimpri and in private sector industry. Chloroamphenicol, isolated in 1947, is a broad spectrum antibiotic. It is rapidly absorbed from the gastrointestinal tract and hence can be given orally in case of typhoid, dysentery, acute fever, certain form of urinary infections, meningitis and pneumonia. Vancomycin and olofaxine are the other important broad spectrum antibiotics. The antibiotic is supposed to be toxic towards certain strain or cancer cells. This is the general structure of penicillin and this is the structure of chloroamphenicol. Next we discuss about antiseptic and disinfectants. Antiseptic and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptic are applied to the living tissue such as wounds, cuts, ulcers and diseased skin surface. Example are furacin, sulfamethoxam, etc. These are not ingested like antibiotics. Commonly used antiseptic Dettol is a mixture of chloroxylenol and teripinol. Bithinol is added to soaps to impart antiseptic property. Iodine is a powerful antiseptic. Its 2 to 3% solution in alcohol water mixture is known as tincture of iodine. It is applied on wounds. Iodoform is also used as an antiseptic for wounds. Boric acid in dilute aqueous solution is weak antiseptic for eyes. Disinfectants are applied to inanimate objects such as floor, drainage system, instrument, etc. Same substances can act as an antiseptic as well as disinfectant by varying the concentration. For example, 0.2% solution of phenol is an antiseptic while its 1% solution is disinfectant. Chlorine in the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide in very low concentrations are disinfectant. Next is anti-fertility drugs. Antibiotic revolution that provide long and healthy life to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food resources, environmental issues, employment, etc. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are used in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthetic progesterone derivatives are more potent than progesterone. Norethindrone is an example of synthetic progesterone derivative most widely used as anti-fertility drug. The estrogen derivative which is used in combination with progesterone derivative is ethylestradol. These are the structure of norethindrone and ethylestradone. So this is all for this topic. Thank you.